Hello everyone, welcome to another video tutorial. So in this video lecture we will discuss about another type of chromatography which is named as thin layer chromatography or commonly known as TLC. So let's start with the introductive part. So before moving to the thin layer chromatography methods, let's understand first that what is the chromatography. So basically the chromatography is defined as an important technique which is used for the separation, identification and purification of the mixture. So the chromatography word is made up of two Greek words which are chroma and graphene. Here the word chroma means color and the word graphene means to write. So at first it was used to synthesize the plant pigments, and that's why it is named as chroma or chromatography. Now the methods of chromatography enables the separation, identification and purification of the different components present in a sample mixture. At first the Russian botanist named as Michael Svet coined the term chromatography in 1906. And after that, the first analytical use of the chromatography was described by James and Martin in 1952. So there are different types of chromatography which are vary in their methods like gas chromatography, thin layer chromatography, adsorption chromatography, paper chromatography and many others. So in this video lecture, we will discuss about the thin layer chromatography, its procedure and its importance in detail. So let's start about the thin layer chromatography. So basically, it is also a type of chromatography technique which is used to separate non-volatile mixtures. So the thin layer chromatography is performed on the sheets of glass or plastic or sometimes in the aluminium foil which are coated with a very thin layer of the adsorbent material, and that's why the chromatography method is used as the thin layer chromatography. So in this technique the components are separated using a thin stationary phase. So here the thin layer is used as the stationary phase. Now the TLC or the thin layer chromatography is defined as the method used for the separation or identification of those components from a simple mixture by the use of finely divided adsorbent which is sprayed over a plate and a liquid solvent which is used as a mobile phase. So basically we know that all the chromatography methods use two phases which are named as stationary phase and the another one is named as mobile phase. So in the case of thin layer chromatography, a very thin layer is used as the stationary phase and a solvent is used as the mobile phase for the separation of different components present in the mixture. So before moving to the procedure of the thin layer chromatography, let's understand on the principle in which this method works. So the thin layer chromatography is based on the principle of adsorption chromatography and the another type which is named as partition chromatography. So here when the thin layer of the adsorbent material which is usually can be silica gel or sometimes at aluminium oxide or cellulose are coated as a thin layer, this layer of adsorbent is considered as the stationary phase and the sample is applied on the plate where a solvent or a mixture of solvent which is used as the mobile phase is drawn up via the capillary action, and by the capillary action of this solvent the separation completed of the mixtures or the component of the mixtures present in the sample mixture. So here the thin layer chromatography depends upon the separation of those compounds according to their relative affinity which is named as RF value towards both of the phases like stationary phase and the mobile phase respectively. And after the separation those compounds are identified by different types of detectors or different detection techniques used by researchers. So this is the principle of the thin layer chromatography. So now let's discuss about the components or the instruments used in the process of thin layer chromatography. So basically the method includes TLC plates, TLC chamber, a mobile phase and a filter paper for the completion of thin layer chromatography. So the first component for this method is TLC plates, which are ready-made with a stationary phase, and these plates are stable and chemically inert plates so that they cannot react with the sample mixtures. Now the another component includes TLC chamber, which is also named for the thin layer chromatography chambers. So these TLC chambers are used for the development of these TLC plates, which are used as the stationary phase in this method. Now the chamber maintains a uniform environment inside for the proper development of the spots of the sample mixture. The third component are mobile phases, which is comprised of the solvent or a mixture of solvent, and there are different types of solvents which are used as the mobile phase in the thin layer chromatography. So the mobile phase is used to perform the capillary action and to move the spots or the sample mixture towards the TLC plates or the stationary phase of the thin layer chromatography. Now the fourth component and the final component of the TLC is used as a filter paper, which is made up of cellulose mainly, and this is the moisture in the mobile phase, which is a solvent, and it is to be placed inside the chamber. So this filter paper is used to help in the development a uniform rise in the mobile phase over the length of the stationary phase. So these are the components used in the process of thin layer chromatography. So now let's discuss about the procedure of the thin layer chromatography. So basically the method includes several steps, and the first step for the thin layer chromatography is to apply a sample spot in the bottom of the TLC plate. So in this method the TLC plate is used as the stationary phase and the sample mixture is spotted in this plate. Now after then the sample solutions are applied on the spots which are made by the pencil, and then the mobile phase is poured into the TLC chamber. Now here the mobile phase can be any solvent or also can be mixture of solvents, and then a moistened filter paper in the mobile phase is placed on the inner wall of the chamber so that it can help in the development of the mobile phase. After then the plate which is prepared with the sample spotting is placed in the TLC chamber so that the side of the plate with the sample line is facing to the mobile phase. After then the plate is then immersed, and then sufficient time is needed to the development for those spots. After then the plates are then removed and allowed to dry for some time. So after the drying these plates, we can see the separation of different types of pigments or molecules present in the sample mixture, and then they can be visualized or can be identified by different types of detectors or detecting techniques used by scientists or researchers. So this is the procedure followed for the completion of thin layer chromatography method. So now let's discuss about the applications of the thin layer chromatography. So basically the thin layer chromatography is very simple, and that's why it is widely used in industries and as well as in different laboratories for the qualitative analysis. So it is used in the qualitative testing of different types of medicines like sedatives, antihistamines, steroids and many other medicines, and they are tested on their qualitative analysis by the method of thin layer chromatography. Now the another application of the TLC is in the biochemical analysis, such as separation or isolation of different types of biochemical metabolites from the blood plasma, urine and different other body fluids, or also from the serum is separated by the method of TLC. TLC is also used in the cosmetics industry. So basically it is used in the qualitative analysis of different types of pigments or molecules present in cosmetics. It is used to identify natural products like essential oils or different types of volatile oils or waxes or alkaloids present in different plants pigments. The TLC is also widely used in the food industry to separate and to identify colors which are used as food colors in different food items or different sweetening agents and preservatives which are widely used in different types of packed food items. Now the another application of TLC is used in the monitoring of the progress of reactions. It is widely used for the monitoring for the progress of reactions in different laboratories. So basically it is a very simple process, and that's why it is used in different types of laboratories and industrial methods. But besides this, there are different limitations of this method also. The main limitation is they can also only used for the qualitative analysis, not for the quantitative. And it can tell the difference between enantiomers and some isomers. So it is not used for those purposes. So these are the limitations of the TLC. So in this video we have discussed about the thin layer chromatography, its functions and its limitations. Thank you.